RNA structure and transcription. RNA. Like DNA, RNA is composed of nucleotides with a sugar phosphate backbone and 4 unique nucleotides. There are key differences. RNA sugar is ribose. RNA is a single chain of nucleotides and has multiple shapes. Instead of thymine, T, RNA has a base, uracil, U. RNA. RNA is essential for bringing the genetic information stored in DNA to the cell, where it can be used. RNA's 4 bases bond in pairs, with uracil replacing thymine. Adenine, A, bonds with uracil, U. Guanine, G, bonds with cytosine, C. U requires less energy to produce, but is not as stable as T. T is better for long-term storage of genetic information, which makes it ideal for DNA. U is more efficient, but exists for a shorter period of time, which makes it more useful for RNA. RNA. RNA molecules can fold and have higher order levels of structure stabilized by hydrogen bonds. As in proteins, the shapes of RNA molecules help to determine their functions. RNA. A strand with the bases in the sequence ACU, AGG, UAC, AUG has a different shape and functions differently than a strand with the sequence CUA, GAA, CAG, UCA, A. Letter changes result in a new shape and new functions. These are illustrative sequences and images of different RNA shapes. Transcription. Transcription is the process by which RNA strands are synthesized from DNA strands. Transcribe means to write out or rewrite. The process of making RNA from DNA is called transcription because the DNA sequences of nucleotides is rewritten into the RNA sequence of nucleotides. The RNA sequence of nucleotides will match the DNA sequence with the exception of U replacing T. The process of transcription is similar to that of DNA replication. Transcription. This is the first step in the transport of the genetic information contained in DNA to the cell where the information will be used to build proteins for the cell's use. DNA has 2 strands. The template strand is used as the template for the enzyme RNA polymerase that will create the mRNA molecule. mRNA will take the information out of the nucleus where it can be used to create proteins. The non-template, or coding strand, is complementary to the template strand and will match the mRNA molecule with the exception of T being replaced by U. Genes. The segment of DNA needed to make a specific protein is called a gene. The genes reside on the non-template strand. DNA is a good archive for genetic information since the bases are protected on the inside of the helix. But this information has to get out to the cell. Transcription. Initiation. RNA polymerase attaches to the promoter region on a template strand of the DNA at the beginning of the gene that it wants to copy. Each gene has its own promoter. The promoter is a specific sequence of bases that the RNA polymerase recognizes. Transcription. DNA strands. This table summarizes the differences between the non-template and template strands. In the non-template strand, it does contain a gene, RNA polymerase does not attach to it, and it is not transcribed, versus the template strand does not contain a gene, RNA polymerase does attach to it, and it does get transcribed. Transcription. Elongation. RNA polymerase runs down the DNA template strand, reading the bases and bringing in the new RNA nucleotides with the proper complementary bases. As the RNA polymerase runs down the DNA, it unwinds the DNA. The RNA molecule being created will be an exact copy of the non-template DNA strand. Transcription. Termination. RNA polymerase gets to a sequence on the DNA called a termination sequence. This sequence signals the RNA polymerase to stop transcription. The RNA polymerase falls off the DNA, the new RNA strand separates from the DNA, and the DNA recoils into a helix. Click here to watch an animation of transcription. Transcription. Just like in DNA replication, RNA is made from the 5' end to the 3' end. DNA replication versus transcription. In DNA replication, 2 new double-stranded DNA molecules are produced. In transcription, 1 new single-stranded RNA molecule is produced. In DNA replication, adenine from the parent strand bonds with thymine on the new daughter strand of DNA. In transcription, adenine on the DNA strand bonds with uracil on the new RNA strand. In DNA replication, the whole DNA molecule is replicated. In transcription, only the strand with the code for the gene is transcribed. Synthesis of both occur in the 5' to 3' direction.